Thank you, Guille and Siddhartha and the organizers of this school. It's a real pleasure to be again in Natal. It is very challenging and very difficult to make a 2-hour presentation talk. So for the students here, one day when you are given, the challenge of preparing a 2-hour talk, you will appreciate what the professors of these schools have to, go through, because it's difficult to stay awake for 2 hours, and I will do my best to keep you awake. So the structure of the class will be the following. First, I'm going to describe in general what the field is about, what the history of the field is about, what the questions are, and then in the second part of the talk, I'm going to be focusing more on the work that we do in the field or in the lab. Some of the people in the schools are working in the lab, like Matias, Santiago, and Flor, and I will be giving due credit when I mention their work. This sentence is very important for everyone working in Birdsong because it's like the starting sentence of all your grant applications. So it's very important that you believe me, and I have to stress this very much, although we'll be discussing a little bit about it. It's a good animal model to study the mechanisms of vocal learning. What I mean by that is that we know that we humans need a tutor. We need to be exposed to a tutor in order to acquire the proper vocalizations that we humans use to communicate. And we share that with a few examples in the animal kingdom. So we share that with cetaceans, a few cases of mammals are reported here and there, but then you have a huge gap until you reach birds. And in there, about 40% of the birds, in particular oscine birds, do share this property that they need to be exposed to a tutor in order to learn the vocalizations typical of the species. By this I mean that if you take a bird that is not an oscine or a hummingbird or a parrot, you can deafen it as a baby and it will generate the typical vocalizations of the species, like a pigeon. In a pigeon, you can deafen the pigeon and it will sing anyways. But with a canary or a zebra finch or a parrot or a hummingbird, you won't have the specific, vocalizations of the species if you don't allow the bird to be exposed to a tutor in a given period of time. Why did I get interested in that? So this is a school about physics and biology. I want to tell you a couple of words about my own background. I did my PhD in Drexel University in algebraic topology. I used to do applied math. Then I evolved into physics, and then I evolved into biology. But always my main interest, if you ask me where I feel comfortable, is in nonlinear dynamics. I tend to think like a dynamicist. So why did I get interested in this? Basically, I started not from the field of neuroscience. I started with the biomechanical question. My question was, well, you have all these beautiful and complex vocalizations, and I was interested in a dynamical question, that is, how much of the complexity of the sound was due to the nonlinear nature of the peripheral system. So my question at the beginning was not involved with neuroscience, it was related to biomechanics. But then I had another cultural question, that is, what do we do when we are physicists and want to approach a problem in biology? Can we do the kind of progressive modeling that we do in physics? It's not obvious at all. I mean, if you talk to a physicist, in principle, physicists love very simple rules, minimal action, okay, very simple rules. Synthesis is like the idea of rigor. And then when you go and try to interact with the biologists are used to evolution. They know there is a minimum level of complexity below which life just doesn't happen. And they don't like oversimplification. So the dialogue is very complex, it's very difficult, and there is no way to overcome that difficulty, because the idea of what is to be a good biologist is not necessarily the same idea, the same concept, the same standard of rigor that it is to be a good and rigorous physicist, right? The idea of rigor is sometimes different. So when I come and I say, well, I'm going to simplify this problem, I'm going to simplify this. To study Birdsong, I'm going to study these few effects. Can I start to do that? Am I going to get something that is pertinent or relevant or not? When I try to model some part of the neural circuit, am I going to be saying something that is pertinent or not? And that's a general question that was very attractive to me in terms of the interaction between physics and biology. So most of the talk is going to be, so how did I evolve after that? I started to work with a biologist, then I created a lab in my physics department. I started to get funding from NIH. Now half of my students are from biology. My committees are from biology. So some of the stories are stories of how physics and biology get mixed in the process of trying to solve a particular problem. And I have many birds in my physics department. We do surgery. We have veterinarians helping us. Yeah, they hate me like hell. So, for example, just to give you the flavor of it, there was a period where people would put signs in their doors, some hardcore, physicists saying, if you are not doing quantum physics, you are not doing physics at all. So, fortunately, I always say, well, we're lucky enough, and Physics Today wrote about us, and then they stopped bugging us. But it's a tension. It's a tension. So landmarks in history. Of course, the study of Birdsong is very old, and in the 50s, Thorpe and Marler had a breakthrough, and it's very interesting. If you think about it, it was a neurological work, beautiful, about the existence of dialects for birds. That means one species that were groups of birds that were separated in space had songs that were slightly different. Okay, and many individuals share one song type, another individual share another song type, and so on. But what allowed them to have that breakthrough was the possibility of doing a quantitative analysis of something they knew about. They knew that there were dialects and there were songs and there were differences, but how do you quantify that? And it's very naive now if you think about it. When they had the first graphic representation of the song is when they started to do quantitative analysis of it. So to have the spectrogram, that is a representation where you have the frequencies of the vocalizations as a function of time, the spectrograph or the sonogram, it is a very naive idea, but allowed to have a qualitative change in the study of the field. So the first systematic work in the field was in the 50s, Thorpe and Marler, and it was mostly about neuroethology, about dialects and behavior associated to different songs. But it was very clear that if you have dialects, that means regions where birds would sing different dialects, then learning was playing an important role in the acquisition of the vocalization. The second wave of understanding this was in the 70s. So Fernando Nottebohm in Rockefeller University identified that in the brain of the Oscine birds, and this is the brain of a canary, you have regions where a few thousand neurons would be highly interconnected, and he made this observation that if you lesion some of these nuclei, then the bird would stop singing. On the contrary, if they would lesion other of these nuclei, the bird would continue to sing, but they would get interrupted in their ability to get new songs. So some nuclei were involved in the generation of the song, and some nuclei were involved in the correction in certain periods of time of the vocalizations. I'm going to be talking about this neural structure in a while and during the talk. But it was very interesting. Vocalizations reflected the operations of a definable collection of neural structures. This is a blessing and also is a curse. Because once that you start to think about nuclei and little boxes that are interconnected, then you are like in a prison, right? It's a conceptual prison. So it's beautiful, but at the same time, it's kind of dangerous. And another breakthrough that was very important in the field was when people were able to monitor the neural activity in awake singing birds. That means here what you have is a bird that is singing, and has some electrodes placed in its brain in different regions, and you can monitor the activity of different neurons while the bird is singing. I'm going to be talking about these experiments a lot in the second part of the talk. But I want to mention other techniques that have been used in the last years to study this problem. A few words about song structure and nomenclature. Okay, this is typically how a sonogram looks. Oops. Like a sonogram looks. All these continuous lines, all these continuous regions corresponds to syllables, okay? And what the bird is doing is creating huge pulses, expiratory pulses of Airside pressure, and through a mechanism that we're going to be discussing, it generates sound. So typically, people study these structures in terms of syllables, and eventually subunits of syllables that are called notes. And this is, for this one species, this typical work of what dialects are like. So with different colors, what I'm presenting are individuals that sing a particular dialect, a particular version of the song. So here in this region, you have the green ones, here you have the red ones, and so on. So birds that grow together tend to sing similar songs. But of course, as you can see, all of them also share some features. Like, for example, all of them are starting with particular notes, and then they combine that with some down sweeps here, and they interpolate with some very noisy sound. So birds do have the ability to learn, but it's not that a species will do, I don't know, a Sonotrichia capensis will not do a Syrinus canaria song. Okay? So there are boundaries of what people can change and can get to produce. Okay, so how does this ability evolve in time? Well, it depends on the species, but typically after the hatching, there is a period where there is a plasticity, a sensory plasticity. If the bird is exposed to a tutor in here, then it makes a representation of what it wants to sound like, and then there is another window where the bird is able to modify through practice its vocalizations. In some species, these periods coexist in time, and in some species, they are sequential. Okay? So for canaries, for example, it's sequential, and canaries have also the ability that every year they can incorporate new songs, while other species only learn their songs once in their lifetime, and once that they learned it, the only thing that they can do is to repeat it, okay, ad infinitum. And this is a figure that I find very important for the whole field, and is slightly forgotten. How can I say this? There are two big books in science, right? It's Darwin's Origin of Species and Newton's Principia. Those are two huge books. For some reason, some people find it very easy to understand either one. And just a few people can find it natural to read both, okay? But if you do something in physics, you have to understand Newton, and if you do something in biology, you have to put it in evolutionary perspective. So this is the brain of an ocine bird, like a canary, that learns to sing. And this is a brain of a bird that does not learn to sing. So this bird sings, and if I play a song by this bird, you won't tell me immediately that it's a non-song bird. It's a great song, beautiful song with different syllables and so on. So what does this guy use? It has a nuclei. Okay, it has respiratory nuclei, because birds first breathe, and then birds learn to sing on top of breathing, right? So it's something that appears after breathing, and you have to coexist with breathing, okay? So when you are breathing, eventually you control muscles in the syrinx, and you generate sound. And when you combine them, you generate song. Then when you have oscine birds, you have a whole collection of nuclei that allow you to modulate that. But that basic structure exists. So these guys in the middle are birds that for all of us in Latin America are very near and dear. Those are suboscine birds, and they're very close to oscines, but they're believed not to be learners. And they have an incomplete structure of modulation between what the birds need in order to sing and what they need in order to modulate the singing, okay? So in this case, in the case of the suboscines, just to finish that part of the story, in the case of the suboscines, behaviorally, in some cases they do it, and they have to establish a way of communicating with the right mate, and therefore in the process of creating the couple, it is very important that they establish a consensus on the dialect. So I think that in this case, for some suboscines, it plays a role in order to generate this duetting behavior with the female. I'm not going to go into the details of all these nuclei, but in the field, there is a tendency to emphasize the top-down approach. That means the telencephalon, with the nucleus that is the high vocal center, is engaging the activity of the robustus nucleus of the archistratum. Then that controls these nuclei that affect the behavior in terms of the positioning of the seedings and the respiration. This is a top-down approach that is based mostly in the fact that HVC is very easy to measure. Okay, so since HVC is very easy to measure, it's very tempting to say that this is the starting of the story, right? This is easy to measure, and I can work in it. Actually, we do work on it a lot because it's very easy to measure too. So people try to emphasize its importance. But of course, there is also all this information from the brainstem to the telencephalon that if you look at it from an evolutionary perspective, it makes all the sense because, again, all this is existing before the appearance of all these nuclei. So it's very important that you have in the telencephalon an information of what's going on at the level of the brainstem in order to integrate that in the behavior of the... And this is, if you want, to add a level of complexity, just to remind you that you have a left side, a right side, and you have all this communication at the level of the brainstem between the left side and the right side. So, again, very simple. How does it work? RAM and PAM are involved in expiration and inspiration. They are generated the respiratory patterns that you need to sing. Then you have N12, that is a nucleus that is going to be innervating the vocal organ. And then you have information that is sent to the telencephalon and is fed back to the brainstem. In here, you have an additional circuitry that is the anterior forebrain pathway, that is the one that is involved actually in the modification of the signals in the process of the learning. This is something that I borrow from Santiago. I don't know where you are, Santiago. It's from his research project. This is typically a typical recording in terms of electrophysiology that you do. As I was mentioning, it's very easy to go to the telencephalic nuclei because they are very close to the scalp, and therefore it's very easy to insert an electrode, typically with an impedance of, I don't know, 3, 4, 5 megohms, and get information from just a couple of neurons, which with some spike sorting you can convert into a single unit recordings. And I'm going to be discussing these measurements later in the talk. Okay, this is the seedings, the vocal organ. So the bird is breathing. Okay, the bird is breathing, and at some point, it will start to generate these downsweeps. In order to do it, it will modify the muscles in here and change the position of this labia. And therefore, when the airflow goes through, it will start to make them oscillate. The modulation of those oscillations is the sound. Okay? So what the bird has to do is to combine the airflow with the tension in this labia, So the total output of the nervous system that I was describing is to generate this coordinated action on these muscles. So when we started to do models of this for the biomechanics, one of the first problems was how to validate them. And in order to validate them, you have to go and measure. So these are kind of measurements that we do in the lab that consist in recording the song and at the same time register the pressure of the air sacs and the activity of different muscles. How do you record the activity of the different muscles? You have to do a surgery. You have to open the skin, open the air sac, go to the muscles, which are very tiny, insert microelectrodes, send the electrodes through a backpack, and do the surgery very fast and in a very efficient way so the bird is not in pain and is willing to sing very rapidly because the electrodes will get out after 3 or 4 days. So you have to do this very fast. And you have all these muscles which are controlling the positioning of the syrinx. And this one is very important, is the syringalis ventralis muscle. It's a huge muscle that controls the distance between the bronchi and the labia are inside that. Therefore, when you contract these muscles, this distance gets larger, and then the labia get more tense. Therefore, they start to oscillate at a higher frequency. So in order to control the frequency of the vocalization, typically you change the activity of this muscle, the syringalis ventralis. So here is the syringalis ventralis that I was telling you before. And the other are the other intrinsic muscles. So I always describe this because it was our first idea. Do these very simple models, of trying to do the biomechanics of the labia make any sense or not. So when we were really starting this many years ago, with Tim Gardner, what we decided to do is, okay, let's generate some synthetic sounds with these mathematical models, and let's see what the birds do. If we expose a juvenile to these songs, can they learn? Can they learn whatever? Which are the limits of what they can learn? Since now we have some synthetic models, if they could copy that, we could explore which is the limit of what they could copy. So we started to generate these very crazy songs, which individually are made of syllables that are more or less like canary, but they are changing in a very plastic way. And we tried to see what the baby birds were doing with that artificial song. So the first surprise was that the birds were willing to take these synthetic songs like tutors. So let's listen to them. So these are very primitive attempts many years ago, okay? But this is a very low-dimensional mathematical model of how the syrinx works, generated synthetic song. So acoustically were things that the bird could do, but in an order and in a rhythm that no canary will ever do. And you had to see all these juveniles practicing so hard for a whole year. Day after day for a year until the spring. So when the spring came, in a week, that sounds like a canary. So the birds were copying beautifully all these alterations of the fundamental frequency, but when the spring came, they knew that they were not going to conquer any canary female. That's exactly my point. So, my point is that there is a very deep interface in between what is programmed genetically and what is being learned. So, at some point, what you are learning is okay, but something genetically is turned on, and something in there, very different and that you were not exposed to, is happening. Well, just to display other technologies people do, well, what happens when the bird is exposed to a copy that is altered about what they are planning to do? So they are planning to do something, and you modify what they are listening to, and you try to see how they adapt and how they can, in different parts of their plastic history, whether they can adapt or not to the changes. This is a technology that is very instructive, and there are many fun stories about this. And this is a story that Matthias is working on, how you manipulate timescales in the brain. This is a technique that was proposed first by Michael Fee in MIT, but we have reasons for which I'm going to be describing in the second part, for which we think it's very interesting to do it in canaries. But typically what you do here is you manipulate thermically one of the timescales that is controlled by one of these nuclei. So since typically these instructions come from the interaction between different timescales along the system, by manipulating one of the timescales in here, what you are doing is basically you are changing the forcing in that problem. And that is something that Matthias has been working on. And this is another technique I'm going to be describing in the second part of the talk. It consists in trying to place electrodes at different depth in the brain. So typically you have a micro drive, and you can place the electrode in the part of the brain you are interested in order to monitor what the bird is doing with different parts of the brain while it's singing. So this was done by Ana Amador, who is a researcher now in my lab, and is the one that has been doing the electrophysiology, the chronic recordings of this in Chicago. I was mentioning the Airside pressure measurements, and I'm going to be doing a lot of description about that. So what this bird is having a plastic cannula that is inserted in a thoracic air sac. This goes to a pressure transducer in here, and you can monitor with the voltage fluctuation what's happening with the air sac pressure while the bird is singing. Of course, another technique, which is a favorite in this field, is doing slices. So the typical work in slices and trying to do the identification in each of the nuclei of which of the neurons are present, whether they are projecting neurons, interneurons, excited or inhibitory. This is done with a lot of care, and Rich Mooney, for example, in Duke, is an expert in this, and this is another technique that is used in the field. So there is a lot of work about what is the behavior of individual neurons in this system. But if you really want to do work in Birdsong, something that is very interesting is that you have no idea the kind of crazy things you have to do if you want to elicit behavior. Because you can do all this beautiful technology, you can do all these motors, and the birds may pay no attention at all unless, for example, you do something as silly as putting a model, okay, that can be a wooden guy, which is painted like this, and it's very sad because you are doing all these mathematical models and these techniques, but then you have to go and paint a little bird and move it around or the bird won't sing, right? Behavior is very complex. It's very complex. Okay, so what it means is the following. This peripheral system is the syrinx, the avian vocal organ. It has two labia in the juncture between the bronchi and the trachea. And these labia, when they start to oscillate, they present nonlinear oscillators. They start to oscillate in one of two mechanisms, either a Hopf bifurcation or a saddle node in the limit cycles, in specific ways, which are determined by the nonlinear nature of the oscillation. These are oscillators that have nonlinearities because when they collide with the walls and when they collide with each other, they have a huge loss of dissipation. And therefore, the behavior of those oscillators is not like the one of a linear oscillator. So the way in which they start to oscillate is not the usual linear oscillator. It's a nonlinear oscillator. And it has a signature in the acoustics that is generated. let's say, for example, when I say a complex acoustic feature, this is the typical song of a zebra finch. It has tonal sounds, but also it has sounds that have a fundamental frequency and a huge harmonic content. So the sound is very rich. Then the vocal tract and the orosopharyngeal cavity modulate the different frequencies in there, and you have the total output. So if you study spectrally what happens with the sound in here, you have a very complex Fourier spectrum. So you need many Fourier components in order to reproduce that sound. But if you take this sound in here, you have only one very tone, pure tone, okay? But it's very interesting if you look. So this is the question. How does the bird control all these different features? Is it a very complex neural architecture that is coding for all these different features or not? Do I have to understand how all this complexity is coded? Does the bird have to learn all these features individually and use some neurons to code for all these features or not? And it's very interesting because if you look at what happens between the relationship between the spectral content index, how spread the spectrum is, as a function of the fundamental frequency, there is a logic in all these sounds. So all these sounds sound very complex, but if you look at many syllables of many different birds, and in all these colors you have syllables from, 25 syllables from, 10, 12 birds, what you have is that they obey a very simple rule. So whenever the fundamental frequency is very low, spectrally they are very rich, and the other way around. And it's very interesting because if you start to work with a very simple biomechanical model of the seedings, and this is what I was meaning by that, you take the labium, okay, as a little elastic piece of tissue, and you write the equations, taking into account the nonlinear dissipation and the way in which the average pressure is established in between these labia. And if you do an analysis of the solutions of that problem, what you get is a bifurcation diagram that is the typical thing that you get when you study the dynamics of a nonlinear equation. Here you have two parameters, the air sac pressure and the tension. And the different regions correspond to different regions in parameter space where the behavior of the labia is qualitatively different. So in here, the labia are quiet, so the pressure is very low and you don't have oscillations. And when you increase the pressure, the labia start to oscillate. The way in which they start to oscillate can be in one of two ways. Either it is a Hopf bifurcation and the oscillations are born with zero amplitude and a well-defined frequency, or they are born in a saddle node in a limit cycle bifurcation and they are born with a huge spectral content and zero frequency. Okay? If you analyze what happens with the oscillations in here, what you get is that the relationship between the spectral content and the fundamental frequency is this analytical curve. So what I mean is that all this complexity, where you have some sounds that are very simple and some sounds that are very rough, they're not coded individually like features of individual syllables. They're just a consequence of the trivial biomechanics that is used by these pieces to generate the oscillations. Okay? This is what I mean by the nonlinear nature of the peripheral system determining some of the complexity of the acoustic signal. And you can go further because instead of working with that biomechanical model, you can do a normal form reduction. Normal form reductions are like the dream tool when you do nonlinear dynamics. How do I know that I modeled the right biomechanics in this problem? It doesn't matter because many different nonlinear systems get mapped to the same class, the same normal form that is a nonlinear differential equation with a minimal number of nonlinear terms presenting some dynamical scenario. So I can, I could have been doing some approximations that are not completely kosher, but it doesn't matter. You don't have to get everything absolutely right. There is something very basic of this system that is captured by this model and is reflected in that when you simplify the model and you write what is known as the normal form, the kind of behavior that you get is qualitatively the same as the behavior of your biomechanical model. But what do you gain? What you gain is that you have a super, super simple thing that depends on two parameters. This inherits the information of the Airside pressure, and this inherits the information of the tension, the synergistic action of different masses that get mapped into the same parameter. Okay, so X is the mid position of the labium, the mid position of the medium, and Y is its velocity. Okay, so what you have is an oscillator, and you assume that you have two modes, a lateral mode and a flapping mode, okay? And X corresponds to the position of the midpoint of the labium. That's right. These are the that's right. These are the ones that inherit the dynamics of the tension and the pressure. Those are things that come from the nervous system, okay? So what we do in the lab is we measure them. And we can plug it in here and synthesize song. And see if the song that you synthesize, when you feed it with the physiological information, corresponds to what the bird was singing. So, for 10 years, I tried to build confidence in different ways for that model. The first one was the following. How does this model assume that you generate the phonation? You start with your parameters, K and beta, in a region where you are not phonating. You get into the region of phonation, and then you get out. If while you are in phonation, you are increasing your tension, the frequency of the vocalization continually increases. So you increase it like this. So one of the ways was, well, let's try to measure those parameters, alpha and beta. And of course, nothing is so simple, but this is a typical recording, as I was showing you before. This is the Airside pressure. These are the EMGs that have to be smooth to generate the K parameter that I was describing before. So in terms of your comment, the shapes of these are what that's right, the alpha and beta. And this is another way to test this. Let's take the model, let's reconstruct from a recorded song how these parameters should be in order to generate something with the same fundamental frequency, and let's see how it sounds. So I'm going to be playing you a real song and a synthetic song that is done when you algorithmically reconstruct the parameters that were necessary according to the model. So let's see if we can listen to these two songs. Let me try to put it at the higher volume. So, okay, so this is the Birdsong song. Okay? This is the synthetic song. It's kind of similar, okay? Sounds well. So, how can we do to validate where this is okay? One possibility is just to take the recordings of what are these physiological instructions and compare with the time series that we reconstructed and compute the correlation. They're correlated, okay? But we wanted to do an additional test. For us humans, that sounds okay. For the time series, they are similar. What do the birds think about our model? So how can you ask a bird what it thinks about your model? How we do that? Right. So you have to take a sleeping bird, and what we can use is the replay phenomenon that we were mentioning in the first part of the talk. So we take a bird, and we know that if the bird listens to its own song, the neurons will respond. If it listens to the song of a friend, it won't respond. If it listens to this beautiful song, it will respond. So we got together with a friend at the University of Chicago, Dan Margolias. Anna was working there. And I said, well, I have this beautiful experiment. I've been working for 10 years in making this beautiful model, and it only has two equations, and this is a biologist, and he says, well, if it's two equations, it won't work. And I said, no, listen, this will work because linear dynamics is wonderful. So we do the experiments, and the experiment is this. You play the Birdsong song when the bird is sleeping, and you record like 20 times. And as I was saying before, you have this beautiful response in here when the bird is listening to its own song. A typical test control is you play the reverse song. When you play the reverse song, all the acoustic features are there, but they are in an order that is not natural for the bird. So the bird doesn't respond at all. And then you play the song of a conspecific, okay, a friend that is living in the same cage, and the bird is not responding. Okay? And you play, of course, that in a random fashion. Okay? But now we're putting also the song of our synthetic model. Wow. I cannot even start to describe to you how embarrassing it is. So Dan Margoliouth is like #1 in this field. He's a good friend and everything. And he was very, sweet and lovely, but, there is no more definite way of failing than this. Okay, so for alpha and beta, what we did is the following. We take the song, and we would take the little pieces of the syllables in, let's say, like 40 milliseconds, reconstruct the fundamental frequency in there, and from the fundamental frequency, we had an idea of what the tension should be like. For the pressure, it was an on and off, and that would be generating, given the shape of the normal form, all the acoustic richness of the sounds automatically, according to the model. And nothing, not a single spike. But what I found very interesting is that this is something that I really wanted to do, and I'm probably the only person in the planet that cares about this. But this allows you to see systematically which is the hierarchy of importance of different things in your modeling, right? So here what I'm changing is the dissipation of the oropharyngeal cavity. So I model that as a Helmholtz resonator, and I change the dissipation in there. So when I change the dissipation, I can make it fade away. So I can progressively include the oropharyngeal cavity or exclude it. And in here, I put the noise that I use to modulate the activity of the muscles. And for the noise too, there is an optimal level of noise where the bird responds a lot. Here is the work as we published it a couple of years ago. 60% of responses. At that moment, we never could go beyond 60% of responses. Now with Santiago and Ana, we're getting a result which I love, and I have no idea what it means. We get songs, synthetic songs, that elicit even more response than the Birdsong song. So it's like the crack of the songs. It's something that they just love, and I don't know exactly what we're doing there. So we just better than themselves. So, it's like they recognize that they are themselves, but they listen to them and they say, I'm singing great, It's like. So, if you put the oropharyngeal cavity, it's a Helmholtz resonator that has only one frequency that it likes, and it's typically, for the zebrafinch, around the 4 kilohertz. So it's more emphasized, the frequency of the 4 kilohertz in the second model than in the first model. This is one of the two changes that I can quantify. So if you look at the spectrum, if you don't put the oropharyngeal cavity, you have basically only the resonance of the first resonance of a tube, and then you have the third resonance of a tube that is closed and opened, that is in the 7 kilohertz. But you have no emphasis in the 4 kilohertz. When you put the oropharyngeal cavity and that Helmholtz resonator, you have something at 4 kilohertz. The other thing is the following. In these frequencies, when you put the noisy, you have a tiny little bit of fluctuation that sounds more artificial if you don't have it. So you have this one actually was, probably this is an example where you have the noise. because it doesn't look so synthetic. But if you're interested, then I can show you without putting the noise. This looks like more like a continuous curve. The reason for which I think it's important, I have a prediction about it, is when you have to create the tension from the activation of many fibers in a very small bird, since you have to add an average, the activity of many fibers, but not that many, you still have a little fluctuation in the average of the activity. My prediction is if you have a larger bird, when you average all these fluctuations, they're going to be fading away. So the importance of the noise is going to be smaller for larger birds and for smaller birds. Okay. But now going to the problem of, if you want, the linguistics of this. We have the impression that we make some breakthroughs in the biomechanics, but we would be really interested in knowing the origin of the shapes of K and P. How do they originate in the nervous system? And it was very interesting because one of the species that we worked with was the domestic canary. So this is the Airside pressure of the domestic canary. And when I first saw this, I couldn't believe it. So for the different syllables, you're using different gestures. These expiratory pulses, so you understand, now each of these expiratory pulses corresponds to a region where the birds are going, the labia are going to be oscillating. So these are slow fluctuations, and each time that you go above threshold, you have many of the high frequency fluctuations of the labia, right? So these are, let's say, I don't know, 100 milliseconds, each of these. And then when this expiratory pulse exists, you have oscillations of the labia in the order of the 1 kilohertz, or something like that. So these are the low fluctuations that correspond to each of the syllables. But when you look at all these shapes, they are not arbitrary shapes. If you come from the nonlinear dynamics field, you recognize that these solutions are the solutions of a nonlinear oscillator which is periodically driven. Let me show you what I mean. Here is an experimental record of a canary song. What I'm showing you here are the numerical simulations of what happens if you take a very simple nonlinear oscillator, like, for example, an excitatory population and an inhibitory population, and you drive it periodically. If you drive it periodically like this, the shape that you get is this one here. If you drive it periodically like here, you get dissolutions, and so on. So the morphology of these gestures corresponds exactly to the morphology of solutions, of harmonic solutions of a driven nonlinear system. And it's not something that is very easy to get, because nonlinear systems can be classified by the way in which the orbits knot themselves and link themselves in phase space. So you have families that are very strict. And this corresponds to the solutions of a very simple nonlinear system. If that was the case, you can take this model and have the amplitude of the forcing and the frequency of the forcing. And I still don't say what these timescales come from in the brain of the bird. But, for example, these are on top experimental records, and these on bottom are solutions of this nonlinear system when it's driven in different regions in parameter space. This was done by Leandro Alonso, who is now working in Rockefeller with Marcelo. The interesting thing is that to go from one shape to the other, what you can do, for example, is to change the frequency of the forcing. So if you change the frequency of the forcing, you go from this kind of solution blue to a solution that is green. So you would go from a solution that goes like this to a solution that looks like this. So what I thought is it would be wonderful if we could manipulate the timescales in the brain and induce these changes, induce these bifurcations in the brain of the bird. So we don't know exactly where the frequencies are, but I assumed that one of the frequencies could be controlled by the telencephalon. This is because there was work by Michael Fee where he cooled HVC and he found that everything goes slower. So at least one of the two timescales is controlled by the HVC or a circuit that includes HVC. But my prediction is that now if we do that, we're going to change from one pattern to another. So Matthias built those devices during his PhD in the lab, and he was manipulating this temperature by building these cooling devices, which are Peltier devices that have these gold connectors here. So you do some craniotomy close to HVC, you place this device, and when you send some current, you cool these connectors. So this is a figure from Matthias' thesis, where now he has this forcing frequency that he can change by cooling. And what he predicts is if you are in a certain region in parameter space, where you would go to. So for example, in case B, you start with an oscillation that is one of the little oscillations that are mounted on a DC current, and when you change, you go to a period 2. This is the prediction of the model, and this is what Matti gets when he cools. So probably, really, there are two timescales which are interacting in the brain. One of them is associated to HVC, and you can manipulate it thermally. Okay? So we're doing this bifurcation analysis experimentally by manipulating the timescales. So this is another set of recordings that Matti made where he gets this normal song and he starts to cool. So let's listen to the cool songs while they are undergoing their bifurcation and breaking into pieces. You cool a little bit. Okay? So you start to break it into pieces. And that would be a very interesting thing if it was in the pressure in the canaries. But now I see all these gestures and these patterns all the time, like in the Sixth Sense movie, right? So now what I'm showing you is the zebrafinch, but now I'm going to the K parameter, to the tension. This is what we reconstructed with the procedure that I was illustrating before. So this was very bland. When we did this, we had no idea of what was the origin of these gestures. What we would do is just to take these little pieces of information and reconstruct the gestures that were needed to synthesize something that looked similar. So these are the kind of functions that we would reconstruct. But if you notice what happens with the system when you, the same mathematical system, when you perturb it with different values of the tension, these are the solutions that you get. So you get exactly the same solution. So what seems to be very complicated, it is the emergence of two timescales that are interacting. So the bird of the brain is not working like a lookup table where details are mapped in 10 milliseconds timescales downstream. They emerge from the interaction of these two timescales in a low-dimensional fashion, which I don't know exactly how it works, and then that's the reason we you know. But here it is. Finally, I want to honor the most dumb of the birds, which are the pigeons. And this is a work that Gogi is doing, and I find it very, very interesting. These two species are as close as two species can be. These are Maculosa picasuro here, and this is, Columba picasuro, and this is Maculosa. These are as close as they can, okay, these two pigeons. They don't learn, and if they have any difference, it's in the peripheral vocal device. So let's listen to these guys. This is a non-learner. Okay, so these two have the same time. If you want, they have the same linguistics, but they have a different phonology, qualitatively different. This guy in here has a very ugly sound. So if you do surgeries on them, what you find is that the left side and the right side, these two originate sound by membranes that are in the trachea. So they don't have two pairs of membranes, they have only two membranes. They're very symmetric in Picasuro, and they're asymmetric in Maculosa. And therefore, when you synthesize the song by doing the biomechanics of the Picasuro, it's very tonal. When you do it in Maculosa, you have all these very rough sounds. So we're trying to explore where you can have a mechanism of sympatric speciation through bifurcations, okay? So you don't need to have a geographical barrier, but you have bifurcation to have speciation. So I don't know, well, they don't have linguistics, that's right. That's right, exactly. Speciation through only phonetics. Okay, and I think that's all I wanted to share. The lessons that I take with me after all this work is that you can find a hierarchy of importance in terms of the biomechanics of the problem. So it is not true that we as physicists are completely blind in a problem where every dimension has, the same hierarchy. We can have a fruitful dialogue where there is a hierarchy of importance of effects, and we can work together, physicists trying to minimize complexity, and biologists that keep us in good shape in terms of not oversimplifying, but there is a hierarchy of importance, and that, through the nonlinearity of the seedings, you can translate relatively simple instructions into realistic sounds. Now, as I was saying at the end of the talk, I'm trying to unveil now whether the instructions themselves are the solutions of a low-dimensional dynamics. But of course, this is the problem of computational neuroscience. How to do that? Where does this low-dimensional dynamics emerge from? What is very interesting, though, is that if you look at them, if you look at the models that I was describing before, they are low-dimensional, but both in the case of the pressure of the canary and the case of the tension in the zebrafinch, they are close to a linear singularity. So it is true that you go to low dimensionality, but you are very close to a region in parameter space where you are very singular. And I think that has to do with an idea that Guille, Marcelo, and so on have been pushing over the years, that you have to be in a region that is plastic enough so you can go to very different regimes, like being close to this double zero eigenvalue in here, but then for the behavior, you collapse to qualitatively different low-dimensional systems. So something along these directions that we're thinking about this problem. And that's all I have. Thank you very much.
First of all, thank you for that talk. It was really beautiful to see so many things come together so well. I really enjoyed it. So I want to ask about whether there is a conflict between your low-dimensional model and the type of very precise spike timing that you showed from that picture. Was it Hanlosser and Fee? That paper was 2004 or something like that?
Right.
So if I remember right, and I'm not a Birdsong person, I just sort of watch from the outside, they had spikes lining up at particular features of these things. So if you're arguing that that entire thing, B, is produced by one setting of pressure and one setting of airflow, why would you need spikes to occur at individual features of that thing that you call a holistic syllable?
No.
I'm not trying to. I've just, it's a question.
No. It's a beautiful question. So we're in the middle of this discussion, a very tough discussion with a group of Michael Fee and Richard Hanlosser. As you mentioned, they claim that in between these, you will have a continuous representation of time. When we were looking for where do this spiking occurs, actually, they don't happen anywhere. They happen at specific gesture significative instances. So I didn't talk about that.
Did you tell him? And
yeah,
what's his response?
Yeah, we're having a huge fight. We're having a huge fight.
Oh, so he does not accept this?
No. So he claims that we didn't measure many neurons, and it is true that we have a small set of neurons. So therefore, I don't make it a big point in my presentations, that aspect of the discussion. But my claim is that, in fact, what he measured was a subset of the things, but there aren't that many in the middle. They're very difficult to get, particularly the phasic neurons, because for interneurons, it's very simple to get many measurements, because you just play the song of the female, for example, and the bird will sing. But phasic neurons are completely silent, and they just spike for like 10 milliseconds during the whole song. So they're very difficult to get. So he got a few, and he conjectured, I have a bunch. We measured a few, and we say, listen, these few are just happening in significative motor instances. So my claim is that then the rest of the dynamics happens when you integrate with the respiration downstream. So we're in the middle of the discussion with Michael Fee and Richard Hanlosser. He claims we don't have enough neurons. I claim he's doing something different. And I don't know, we have to measure more.
Well, eventually the data will settle it one way or the other.
Exactly. Yeah.
Gabo, there are African gray parrots.
Huh?
African gray parrots, right? So that they actually, they have a amazing vocal learning ability. It's just that they are difficult to study because they are expensive and protected, but
yes.
They have the same anatomy and they do amazing things.
Yeah. Now, this has been a dream animal system for people because a zebra finch is worth $5. They reproduce in an aviary all the time. And it's not even like canaries that you have to wait, to the spring to have the babies and so on. You put a male and a female zebra finch any time of the year, and after a while you're going to have babies. So it has been a great, inexpensive, beautiful, easy to work with animal. These zebra finches are very tough animals. I mean, it has been great. But to which extent you can pursue this analogy is debatable, if you want. For me, a beautiful thing is to know really how little is learned. How little is learned.
Okay, so let's thank Gabriel again.